Did I leave off here? Like right here. Right here. Okay. So this is a visual of mitosis. It's important that you understand this visual because your projects, your last quiz grade is going to be a project on creating a visual representation through animation of mitosis. Really of the cell cycle, but primarily cell cycle with mitosis. So there's 3 parts to mitosis, which is interphase, mitotic phase or mitosis, and cytokinesis. We kind of already learned that, so that's technically a review. This is what an animation of mitosis would look like. So there's interphase, prophase, metaphase, anaphase, telophase. But of course, in my class, you're not going to just do a simple animation. You're going to theme your animation, and we'll talk about that in a minute, okay? So prophase, metaphase, anaphase, telophase, cytokinesis, or you could say interphase, PMAT, which is prophase, metaphase, anaphase, telophase, cytokinesis, okay? So as a review, mitosis happens in what kind of cells? I kind of heard it. Which cells? Look right here to remind you. Body. Do anybody remember what body cells are called? Starts with an S. Somatic was a good start. Somatic. Somatic cells divide to replace cells that have died or are like going wrong, and to prepare for growth, for your growth. In order for you to grow, your cells have to divide, okay? These are lung cells, skin cells. All these cells in your body, other than sex cells, are considered somatic cells. These are the parts you need to know for mitosis. Let's see if you could remember what they are. I'm going to kind of zoom in. What's this right here? What's this whole picture of? A cell. What's in the center of eukaryotic cells? Nucleus. What's inside the nucleus? DNA, chromosome. If you see the word chromatin, it's the same concept as chromosome, okay? It's kind of like pieces of chromosome, let's say. What's this right here? Centrioles, good, because we already covered them. All right. Then this area here that's blue is the cytoplasm. What is this yellow X? Chromosome. So again, you have chromatin, which is like pieces of DNA, pieces of protein, and then you've got the chromosomes, because it's the way you see things under the microscope that matter. Then what's this line here that's coming out of the centrioles? Spindle. And then, anybody know what holds the chromosomes together? We've never talked about that, but out of curiosity, you know? Centromere. Good job. These parts are going to be visible in different parts of the cell cycle, and sometimes not visible. But these are the parts that you're responsible for referencing on the test and on your projects, okay? So, mitosis happens in 4 parts. What are the letters? PMAT, okay? What does PMAT stand for? What's the first one? Prophase, second one. Metaphase, third one. Anaphase, fourth one. Telophase. So prophase, metaphase, anaphase, telophase, or PMAT is the mitosis part of the cell cycle. You got interphase first, then PMAT. So what is this little thing? Centrioles. What is this little thing popping out of the centrioles? Spindle. So the spindle starts to form and it comes out of the centrioles. What is this red X? Chromosomes. The chromatin coils and chromosomes become visible. This is all prophase. Okay? I should have said that first. And then what's this? Right here? Nucleus. the nucleus is actually gonna disappear and the nuclear envelope starts to break apart. But I want you to look at this right here. Stop writing and look at what I'm about to tell you. This is interphase. During interphase, and we already covered this, the centrioles are together. The nucleus is fully visible, and the chromatin is condensed and coiled all in like a spaghetti-looking mess, okay? During prophase, centrioles move apart. Spindle starts to pop out of the centrioles, and the nuclear envelope breaks, nucleus starts to disappear, and the chromosomes become visible. It doesn't happen from one to the other. You could look under a microscope and see centrioles maybe like almost together and spindle forming between them because they go from being together to moving fully apart. The nuclear envelope goes from being perfect to slightly breaking to fully breaking apart. Yes? It is a process. And I say that because when you look at the animation, and we'll look at this one, I'll show you right now. This is interphase right here, starting. You see my centrioles moving. So they don't just jump apart. They flow apart, okay? So for every part of the cycle, I'm going to give you the previous cycle at the bottom so you can reference what it looked like in the previous phase and kind of say what should happen. So yes, you're responsible for each part of the cycle. You have to recognize these things. So I'm going to give you a question and you're going to have to identify the parts. I'm going to give you a picture of a phase like right here. And you have to know this is prophase because the centrioles moved apart, the nuclear envelope broke apart. Okay? And then when you jump to the next one, here's prophase. Now look at prometaphase. That's kind of like an in-betweener. And it's basically the same thing. I just want you to realize you don't go from one thing to another. It is a process. So by prometaphase, what do we not see here at all? The nuclear envelope. Do you see the spindle fully forming? Yes? Your centrioles completely moved apart. And really, technically, the nucleus is gone because you will not see that dark, large organelle that you see sometimes when you look under the microscope. It's like gone, okay? With a good microscope, you'll see the chromosomes. The one thing I didn't mention that I uncovered is this, in between the chromosomes, there's little dots. Make your little dots visible. Change the color. Make them like orange or something. Centromere. And if you want to change the word to orange so that it makes sense, or black or something, they hold those chromosomes, which can be called sister chromatids, together. Chromosomes, chromatids, chromatin, on the test is all going to be the same thing. Okay? I'm not going to try to confuse you. All right, what comes after prophase? What? Metaphase. We're going to underline M for metaphase. Why do you think I'm underlining the M? What do you see happening? Middle. The chromosomes line up at the equator, which is the middle. Now, it could be the middle vertically, it could be the middle horizontally, depends on which way the cell is facing, but the chromosomes line up at the middle. What else do you see happening that's different from here? You see it right here. That already happened, right? What's connecting? I heard. The spindle connects to what? What in the center? Do you see, notice how they connect in the center? What's in the center? Centromere. Now there's a lot of C words. Make sure you draw your little centromere so you know that the spindle is connecting to the centromere, okay? You got centriole, you got centromere, and you got chromosomes, which are chromatin. When you see centrioles, I'm going to show you this, which I didn't show you before. The centrioles often do like a cross, or they look like this, which has the T or the L word in it. Here they cross like a T, and here they kind of look like an L, centriole, okay? My centromere, underline the M for middle. It's in the center of the chromosomes. So way to remember them. And the chromosomes, you have to know, you've known this for a long time. They're the X-like structure, yeah? Inside the DNA. Inside the nucleus. All right, what comes after metaphase? Anaphase, okay? PMAT, okay? Look at metaphase here. A for anaphase, A for what? Apart, away, good. My chromosomes are being pulled apart. The spindle actually pulls them apart, and the centromere is going apart. I don't know why I put that on both ends, but chromosomes get pulled apart because we got chromosomes on both ends. It could say pieces of chromosomes or chromatid. It doesn't matter. They're getting pulled apart. They were together all down the center of the cell or down the equator of the cell during metaphase for middle. And then they get pulled away or apart. So you could put away or apart for anaphase. What's the last one? Telophase. Now notice, anaphase has one part, metaphase has two little parts, but prophase and telophase have a lot. So telophase is almost opposite of prophase. And then one extra thing. So what's happening? Here's anaphase, here's telophase. Tell me what's happening. What's back together? Centrioles back together. What disappears? Spindle. What reappears? The nucleus. And let's add here too, let's add not just the nucleus, but the chromatin, right? Because I don't have chromosomes anymore. But it doesn't matter. It's still chromosomes. And then I added the cleavage furrow forms. This is your cytoplasm bending. Now I always say T for telophase, T for telephone. That's what a telephone used to look like. You used to have an earpiece and a mouthpiece. You remember those phones? Did you ever use one? A telephone with the earpiece and the mouthpiece? Some people don't. Telophase. What happens after telophase? Cytokinesis. C for cytokinesis, C for the division of cytoplasm. People are getting it. Yes. All that happens during cytokinesis is two identical daughter cells form. Let's put that. Two identical daughter cells . Of course I spelled that wrong. All right, try to label these right here. Okay, I left these blank on purpose. See if you can label the phases. Don't label the parts, just label the phases. Yep, I will. Hold on.